Welcome to Biocomputation with Max. Today I wanted to talk about another bit of biology that's very commonly examined in development of drugs, namely toxicity to the liver. Uh, um, there are physical methods to measure and predict it. For example, this tool from Liana Biosciences called Perfusion Pal has several well-established applications, one of which could be called Liver Pal. Let me briefly show you how this instrument works. There is a three-dimensional scaffold with different cells in individual wells, and media is able to perfuse up and down by a couple of millimeters, providing multiple volumes of exchange daily and oxygenating the medium very richly through oxygen exchange with the liquid blood substitute shown in purple here at the bottom. The pumping action by the pump is reciprocating back and forth, which leads to the media going up and down in the wells. As the picture here shows, the 48 well plate here is able to provide complete isolation of individual well reagents, which is highly suitable for working with the secretome of these metabolizing cells like hepatocytes. Here's a cross section showing the medium, the blood substitute, which is providing this remarkable liquid cushioning. It's a perfluoroalkane, which has almost 100-fold times oxygen content, is twice as dense as water, and is immiscible. With it, it's never in touch with the culture. It's merely acting as a liquid and oxygenating piston as it moves media through the SeedEZ scaffold, which is 0.5 millimeter thick, very fine fibers, which I'll show you next, that house the cells. So here's a culture over several weeks showing 4 and 10x magnification of individual cells that are attaching themselves to this three-dimensional scaffold, which is often coated with Matrigel as an extracellular matrix, and then the cells begin to merge, but there is still open space for movement of media, and it's a pseudovascularized culture, regardless of presence of epithelial cells. You can see that the nuclei are co-located and how the fibers reveal themselves after several weeks. In the future slides, I'll talk about the differences between 2 and 3 dimensions with a plate, which is traditional, or 3-dimensional plate where cells are suspended in the gel, as is typical when working with organoids, or this perfusion pal plate in either static or perfused condition. And the results are from a publication last fall about these cultures with hepatocytes, including primary human hepatocytes. So that's the key advantage. Looking at typical 3-dimensional plate or perfusion pal in a static or perfused condition, we see that the perfusion pal has significantly higher respiratory metabolism across 2 main hepatocyte cell types, HepG2s and HepRGs. In breast cancer carcinoma cells, we see the same. And of course, perfusion adds to the efficacy of oxygenation and therefore a respiratory metabolism, which is very important for cancer as opposed to glycolysis, for example. Here, one of the cytochromes is also showing enhanced activity as we move from 2 dimensions to 3 dimensions, especially with perfusion. Another metric of activity of primary human hepatocytes is production of albumin, which is the most common protein in serum. So here you can see that going from 2D to 3D is beneficial. There is a little bit of improvement on perfusion, but it depends on flow rate. At somewhat higher flow rates, we see more efficacy. So it depends on the specific conditions. In terms of activity of either cytochromes, on this logarithmic scale, we can see dramatic difference between the two extremes, the traditional flat plate and 3D perfused. So this is multiple fold of difference. This is about 30-fold enhancement. In terms of induction of these cells with typical inducing reagents or media, we can see here that 3D perfused, especially for some cytochromes, the 3A is one of the critical ones, is very significant for the perfused case. So the activity is more realistically mimicked with not only 3 dimensions, but this biological stimulation, which is synergetic with that type of culture that mimics in vivo conditions better. The gel that I mentioned previously, Matrigel, actually has a suppressing activity, as you can see from going down from blue to red and then from green to purple. In all these cases, the presence of Matrigel suppresses activity in two-dimensional with two-dimensional reagents, or those that are optimized for organoids in 3 dimensions. So there is not a huge difference. In fact, there is consistency between a response here. What we can see is that 3D plate certainly is generally better than 2D. So the problem can lie with this extracellular matrix coding. In terms of metabolism, this is another way of looking at it. How it's suppressed basically with Matrigel. So there is an alternative, and it's composed of very well-defined chemically components. They produce this extracellular matrix that can be fine-tuned, and kits include ability to not only change the composition of laminin versus collagen versus fibronectin, charged versus neutral, but also adjust the proper stiffness. For example, brain is a very soft tissue as opposed to cardiomyocytes, for example, another muscle group. So this is an alternative that we provide and can be utilized together with primary human hepatocytes that I mentioned. One of our partner companies, Sites Biotechniques, is specialized in delivering fresh human hepatocytes. They also deliver them frozen, but this is a very efficient way of studying liver biology. It's comparable to price and greater in ease compared with induced pluripotent stem cells. So this is a very favorable third reagent. Now, talking about the effect of this perfusion, you can see differences of glycolysis here. So for example, more glucose is remaining in the 3D perfused medium, which suggests oxygenative metabolism rather than glycolysis, as happens with hypoxia. So this is very important in the study of cancer, but also impacts metabolism, of course. Importantly, the production of reactive oxygen species is not affected. So this is a favorable platform for oxygenation. And finally, this panel shows actual measurements with electrochemical oxygen probes of oxygen level. In normal 2D or 3D plates, oxygen level is depleted very quickly from normoxia to severe hypoxia. That affects metabolism going glycolytic. Perfusion cycles, as shown here, are able to keep oxygen content up. In fact, it's possible to program this at a higher frequency so that the oxygen can be maintained. So in summary, this model combining highly oxygenated three-dimensional scaffolds with the right type of matrix and cells can be an efficient means of biocomputing liver toxicity or liver efficacy also, for example, in NASH. Thank you. See you next time.